Long-read genome sequencing. So what is genome sequencing? Genome sequencing is a process where the complete DNA sequence of an organism is determined. So the ability to read and analyze the entire genome has become a revolutionary and crucial development that opens the gates for many applications in modern medicine and biology. Being able to analyze the DNA instructions can allow us to better understand the role of genes, identify and diagnose disease, study evolution, as well as personalizing medicines. Before long-read technology, there were many challenges faced with genome sequencing. The genome is a long and complex structure to organize. It is comprised of many chromosomes, which each contain an extremely long DNA molecule containing many genes and millions of nucleotide bases. The evolution of long-read genome sequencing started with the Human Genome Project in 1970s. So the first gen Sanger sequencing was first used, although this was accurate, it was both time-consuming and expensive. Then around the 2000s, the next generation or second gen sequencing, such as Illumina, allowed us for massively parallel sequencing and higher throughput while making it more cost effective. However, both methods had a very short read length because of needing to first fragment the DNA, which limited sequencing to a maximum of only a couple hundred base pairs in length. This meant that the sequencing of very large genomes, such as the human genome, was time-consuming, and there were also issues such as struggling to organize more complex or repetitive regions of the genome and identifying larger base modifications and mutations. This brings us to third-gen long-read sequencing, such as Oxford Nanopore. So how does this work? Long-read sequencing in general refers to the single molecule direct reading of the DNA without needing for excessive amplification techniques, allowing for real-time sequencing along with very long read lengths of thousands of base pairs or minimizing DNA fragmentation. This increases the accuracy when reading large structural variations and saves time. Specifically for Oxford Nanopore, a long chain of single-stranded DNA or RNA is passed through protein nanopores. As each different nucleotide passes through the pore opening, it disrupts the ion currents across the membrane in which the pore is embedded. Each nucleotide causes a different variation of disruption, which gives off a signal, and this signal is then matched to a specific nucleotide base, which allows the entire chain to be sequenced in one go in real time.
Like most people watching this, you're probably wondering how long-read sequencing technology, such as Oxford Nanopore technology, works. There are two key steps, the library preparation and the sequencing itself. Please note, whilst we use Oxford Nanopore technology as today's model, there are other types of long-read sequencing technologies also available. For example, PacBio's single-molecule real-time sequencing is another mechanism which utilizes DNA polymerase and fluorescent pulses on difficult-to-sequence A to T, G to C repeat-dense regions. In our case, Oxford Nanopore technology relies on changes in current of its pores when different bases go through them. Recall that short-read sequencing seldom is limited because it takes a lot of time and is restricted to short fragments. Of course, Oxford Nanopore technology has its own limitations, which we'll cover later, but often short-read Illumina is combined with long-read Oxford Nanopore technology to holistically screen genomes. In the library preparation phase, samples are prepared. A recommended mixture of approximately 500 base pair oligonucleotides and 5,000 base pair fragments are used. The former are referred to as baits, which help with the specificity of the sequencing. Meanwhile, the latter are your target sample fragments. PCR is optional, but not necessary. Protein conjugated adapters are ligated on by DNA ligase, mainly for identifying different sample fragments. Finally, tethers are attached with motor proteins and are also added to help guide the sample fragments through the nanopores later. Moving on to the sequencing process, we are introduced to the Oxford Nanopore technology flow cell. This is covered in rows and rows of nanopores that are typically graphene nanopores, embedded in synthetic polymer membranes that are electrically resistant. Then a potential is run through the system, thus establishing a current flowing through the nanopores. This is crucial for sequencing. Motor proteins found on the tethers of the fragments attach the strands of nucleic acids to individual nanopores. Then, the fragments, regardless of length, are pulled through the nanopore aperture. Each time a base of nucleic acid passes through, it causes a disruption in the current flowing through the pore, producing a characteristic squiggle on the automated reader. Each of these squiggles are unique to the nucleic acid's bases since all the bases are effectively different. Thus, after successive bases pass through, these squiggles can then be converted into DNA or RNA sequences, since base calling algorithms and technology utilize raw electrical data from the current changes.
De novo genome assembly is a process in which randomly sequenced fragments of a sample are reconstructed to determine the sample genome. The overlapping sequence fragments are stick together to form contigs or contiguous sequences. Ideally, there should be one contig per chromosome. In the technology of short-read genome, they have been problematic in determining mammalian genomes, typically the human genome, usually resulting in hundreds of thousands of gaps. This is due to the repetitive sequences in these regions that are much longer than the maximum git length in these technologies. For example, centrometric DXS1 array in the human X chromosome they are approximately between 2.8 to 3.1 million base pairs, contains about 1,408 copies, with each of them has around 2,057 base pairs. Short-gate technology has a maximum gate length of only 300 base pairs, whereas the long-read technology, typically the Oxford nanopore technologies, can achieve a gate length up to 4 million base pairs. Another application is the rapid determination of infectious disease genome. The process begins with RNA extraction from potential sample sources such as the saliva of clinical patients. The extracted RNA is converted to complementary DNA. The resulting material is amplified to generate the overlapping amplicons, which are then purified, barcoded, and transferred into a main ion nanopore sequencer. This is an Oxford nanopore technology device. After sequencing, the raw data undergo base calling to convert the electrical signal into nucleotide sequences. The multiplexing to separate the data from different samples because the device can run multiple samples at the same time. Filtering to remove low quality data, alignment to the COVID genome, variant identification, and finally, generation of the viral genome consensus. This process can be accelerated to within a single day, making it become critical for vaccination development against any infectious disease right now and in the future.
Although Oxford Nanopore technology is a revolutionary long-read sequencing technology, it still has some limitations. These include the lower raw read accuracy compared to short-read platforms, high computational demand, and the variability in flow cell performance. It also requires high-quality DNA or RNA, which can be a challenge with degraded or clinical samples. To overcome these issues, several innovations are in progress. First, improved chemistries and nanopore designs are already increasing sequencing accuracy, with some kits now reaching over 99% per read accuracy. Also, AI-powered base coding algorithms are reducing error rates and enabling more reliable variant detection. Third, developers are working on more stable, reusable flow cells to improve output consistency and reduce costs. Fourth, cloud-based analysis tools and simplified bioinformatics pipelines are making data analysis more accessible. And finally, Oxford Nanopore technology is moving toward clinical-grade standardization, with efforts to validate workflows for diagnostics and pathogen detection. In conclusion, while Oxford Nanopore technology is powerful, its limitations in accuracy, cost, and accessibility are real. Fortunately, ongoing advancements are quickly addressing these issues, paving the way for broader use in research, clinical, and field-based applications. That's all. Thanks for watching.